eventually you end up with an abnormal number of chromosomes, either too few on one side or too many on the other, because they're traveling together. So this is one of the, non-disjunctions, one of the most common causes of aneuploidy. Okay. Mitotic non-disjunction, is this whole organism or cell-by-cell basis, like a tissue level?
Unless it happens.
Yeah, so it's a little tricky, but typically you're going to think of mitotic non-disjunction being isolated. The aneuploidy that results is going to be isolated to a few cells that it's happening within. So you're going to have aneuploid areas more in the body, not the entire organism necessarily. Okay, so for the most part, you'll have a little small area that every subsequent cell division of that first cell that screwed up and had non-disjunction, every daughter cell after that will show a reflection of that non-disjunction event. Where might it be? Is that a question or your?
And I haven't heard disjunction whenever you those two sister chromosomes separating.
So disjunction could be two homologous chromosomes or sister chromatids separating. Or you could have it in a whole tetrad. When we get to meiotic, you can think about, so non-disjunction can occur, right? This could be either or in terms of disjunction. And meiotic non-disjunction can happen either at meiosis 1 or meiosis 2. So then you're either talking about homologous, like a tetrad, that all four arms go together, all four sister chromatids travel together, or at meiosis 2, where you just have disruption of the way it looks for mitosis. But I think what Carl was about to speak to was, where would we see aneuploidy in the entire organism from a mitotic, or almost nearly the entire organism from a mitotic non-disjunction event? Yeah, during embryogenesis, when you go from, okay, you have fusion, fertilization of sperm and egg, you have one cell, first step is to divide, right? Make 2 cells, then 4 cells, then 8 cells. If at that first, and those are all mitosis divisions, right? So if that first cell division, or even the second cell division, you have non-disjunction occur, the whole organism might be reflecting this event. Okay? Does that make sense? So if this happened very, very early during development. But for the most part, we think of it being areas, isolated areas in the body. And that's in comparison to meiotic, which means your whole organism is aneuploid. So the result of the entire organism, because it was derived from gametes that were produced from a non-disjunction event. So essentially every cell in your, if you took an individual who has trisomy 21 and aneuploid number of chromosome 21s, right, every one of their cells will show that third segment of chromosome 21. They're all going to be aneuploid because that happened in their parents at meiosis during gametogenesis. For the most part, non-disjunction is a fairly random, so the kind of the causes of it, it's somewhat of a random process, so spontaneous. Although we sent, as we mentioned earlier, with the old eggs thing, we tend to see this in aging cells more often. So you tend to see mistakes happening in aging cells. You tend to see it happening more often in cancer cells too, right? So it's cells where checkpoints and basic cellular processes have started to degrade to a certain extent for whatever reason. But in terms of the molecular cause, it's almost always non-disjunction is due to you're disrupting the ability of microtubules to polymerize. So somehow you're interfering with the normal ability of those spindle fibers to grow out and attach to chromosomes. kind of a major failure of a basic cellular process, right? Because we need microtubules for lots of things, including cell division and properly aligning our chromosomes and all of that. So when this happens, you have lots of other things going wrong as well inside of the cell. But generally speaking, we can track non-disjunction events. One of the major causes is somehow you're screwing with basic microtubule polymerization. And this, of course, you can actually induce this in the lab. So spontaneously, this would be in nature or in vivo, right? So naturally, in vitro or experimentally. I spent about 8 years doing this constantly to cells. So you can induce this disruption of microtubule organization, right? You can use chemicals that are called mitotic inhibitors. I think your book uses colchicine. There's a bunch, colcimid. These are all derivatives of colchicine. Colchicine is actually also used as an anti-cancer therapy. Why would that work? Why would you use something that disrupts microtubule organization for cancer therapy? Yeah, so then you're really inhibiting all cell division, but most cancer therapies are really detrimental to the rest of our tissues as well. But this would be a way to stop your cells from dividing, to prevent them from dividing by cutting down that microtubule polymerization function. So these kind of chemicals, they bind to tubulin. They prevent tubulin dimers from binding to each other. So all those karyotypes that I showed you guys and pictures of condensed metaphase chromosomes, folks have added things like colcimid and colchicine to their sample, and they added at a point where what they're hoping is any cell that's starting to condense down, line up at the metaphase plate, the most condensed point, then if you add this chemical right then, you break those microtubule fibers, they can't pull those chromosomes apart to anaphase, and your cell's arrested at metaphase. Okay, so it's kind of stuck in metaphase, and then you can put it on the slide and observe it. So we used to do this with chicken embryos. You just take an egg that's some point in development early on, cut open the top of the shell, inject it with a bunch of this chemical, put the shell back on, let it keep incubating for an hour, take it out, chop the head off, squish it up, and you have a bunch of metaphase cells. So it's kind of a nifty thing to do, minus the chopping the head off part. But the reason this works so well in chick embryos, why would that be? What's happening in the embryo at that time? Massive cell division, right? You have in any kind of growing organism, especially at that early embryonic stage, you have cells dividing like crazy. So your chances, your probability of capturing cells that happen to be in mitosis, in metaphase, are really high. Yeah. If you take cells out of your own brain right now, right, your chances of capturing anything and undergoing mitosis and metaphase are really null to zero, right? Those are cell types that hang out in G0 and don't really go around the cell cycle. Okay. So in an adult organism, you have to be a little more creative about what tissue type you take to get a whole bunch of metaphase chromosomes. Other piece, any questions up there? Another piece about this random spontaneous fit is the numbers actually do support that this happens. So we wrote earlier that this can happen in meiosis, and it can obviously happen in meiosis 1 or meiosis 2. The numbers are much higher for frequency in terms of non-disjunction in meiosis 1. Okay? So you tend to see more non-disjunction at that first cell division. And the reason here, intuitively, should kind of make sense because you have sister chromatids that are attached to each other. Okay, and then you have some kind of recombination crossover event that's holding the tetrads together. So you have a lot of intermingling nucleic acids and proteins and all these pieces and parts to make the machine work. So the more pieces and parts you have, the more likely it is that it'll break, right? And so that, what often happens with non-disjunction and this whole tetrad moving to one side of the cell or the other is the proteins that help resolve that intermingling of nucleic acids, the recombination, the ones that are going to get in there and help separate and make sure everybody is on the right side and you don't have a dangling piece of chromosome. Those can often be either mutated or disrupted or not in the right place at the right time. These are places where we see errors happening in non-disjunction is the result. Okay. So it's whenever, for the system to go correctly, you need that tetrad to be intact initially, right? And then you need it to be able to be separated, that crossover to be resolved. So if they're still hooked together, often we see them get pulled to one pole or the other within the cell. So this is kind of the level that I want you guys to know, understand about non-disjunction. You should be able, and I would encourage you, go home and draw out what the product of non-disjunction looks like at meiosis 1 and what it looks like at meiosis 2. You know, this is again reinforcing, do you know mitosis? Do you know meiosis? Can you draw these chromosomes while you're half asleep on Halloween night or something? I don't know. Go home. I would really encourage you guys, draw that out, show what your products would be, what your resulting cells would look like. So do it for mitosis, which looks the same as meiosis 2, but I would definitely do it for meiosis 1 and meiosis 2. Make sure you can understand and follow that process.